It's lovely to see a good number here. I'm Tom Connor. I'm a senior lecturer at Cardiff University, and as Mark already mentioned before, coming to Cardiff, I was a postdoc at the Sanger Institute in pathogen genomics. So my background is as a pathogen genome researcher, and I guess Nick and I have borne the brunt of a lot of the actually getting the system that I'm going to talk you through now up and running and working. Starting off with some acknowledgements, we've got the whole consortium, and it's not quite a cast of thousands, but it is a reasonably substantial number of people who've invested quite a lot of time in getting the system that you're going to get to play with today up and running. In particular, tomorrow we've got a set of talks, and there's going to be a load of academics talking about things they can do with the project and things and their research. But really, this system that you're going to be using only works because of the sysadmin team that sits behind it. So there's been a lot of technical effort on the part of the sysadmins and who are associated with the project. And I just wanted to highlight them. So we've got Simon Thompson and Marius, and Marius is here at the back, who have been really central to actually get the system up and running. And we're very glad now to have Radislav, who you'll be meeting, I'm sure, over the course of this afternoon, and Matt Bull from Cardiff, who are very active, and actually they've been active in getting the system up and running and stress testing it over the last few weeks. And so while you're going to be hearing a lot from the academics, and I'm sure the academics will be taking a lot of the credit, a lot of the hard work has been done by these guys. So I just wanted to flag that up first of all before I get on to talking about the system. So the system itself, it's an MRC funded project. The project is around about £8 million in size. Approximately half of that is computer hardware. And we basically wanted to build a system that allows us to support a large number of virtual machines simultaneously across our 4 sites. And I guess the tagline is this should be providing a core national infrastructure for microbial bioinformatics. So that's kind of what the task we set ourselves. And so the background to that is this thing that at the Sanger they call the sequencing iceberg, and it's this relatively simple fact that as any biologist who's in the room will know, it's relatively cheap and easy to generate lots of sequence data. It's also cheap and easy now to generate lots of other high throughput data, be it proteomics, metabolomics. It's very easy to do now. But the real costs that come with that aren't in the generation of data, they're actually how you handle that data. And there's a whole host of interlinked facets of being a data intensive researcher which aren't really covered by the cost of the sequencing, and actually are far more significant in terms of things like getting access to compute to actually analyse that data, how you store that long term, and how you have the expertise in your group to actually be able to deal with that data when you want to analyse it. And so the problem that you get in a lot of places is what people in the IT world call shadow IT. And that's the idea of servers sitting in closets. So you've got, you're in a university like Warwick or like Cardiff, there are large central IT resources and central IT teams that run data centers and rooms filled with servers. And yet as academics, quite often what we'll do is we'll slap a server on a grant for a few £1,000s, we'll buy it, and it will probably sit in a closet or in a LAN room somewhere within our academic school. And that's rational, because everything that you do, if you're going to be doing anything with sequence data, for example, is underpinned by having that compute capacity. And I've been in the position, so when I joined Cardiff in 2012, I came from the Sanger, and so I came from this environment where there was a massive 16,000 core compute cluster that was available to everybody on demand, to a university which is slightly less functional in providing bioinformatics support to researchers. In fact, I was the only PI in a department of 120 academics whose first interest and first focus was bioinformatics. So I had to work out how I was going to get all my software up and running, how I was going to develop a local environment to carry on doing my research in this new place. And I did that pretty much on my own. I didn't know where I could install my software, I didn't know what resources were available. And so I had a startup. And so my solution to this problem was to build my own system. So I took my startup, I bought myself some servers, I configured a high throughput computing cluster with some storage, and that's not an unusual story. I know of lots of other bioinformaticians who've been in exactly the same position. They go to a university, they find the systems the university has on offer really aren't suitable, they develop their own system. I know lots of PIs that when they move into doing genomics, they decide they don't have adequate compute capacity, so they buy a server and they have a group server. And so this is not an unusual story. And I think this sort of shadow IT, this purchasing of servers, is sort of compounded a bit by the way that we do things in biology. As a discipline, we tend to think of things like labs, because a wet lab is a physical space, right? So you have a physical space with your name on the door, and you're responsible for the health and safety in that place. You're responsible for getting the equipment into that space for your lab group to do things with. And we often think also of sort of experiments as being wet work. And the problem is that when you take that mentality and you translate it to IT infrastructure, it's pretty easy to see how you can go from thinking of a lab as a physical space and having a piece of equipment in the lab like a PCR machine, with having a server associated with that lab as well. And the problem is, in an IT sense, that's a really bad call, because there are a whole number of reasons operationally why you wouldn't want to do that. And having in a lab an instrument that's not highly utilised, is a bit of a shame. But when you've got loads of labs doing that with servers that are continually costing money when they're drawing down the university's power supply as they're sitting there doing nothing at all, it's a huge waste of money and resources. The other problem with that is if we've got one of these local servers, then we will tend to develop all our software on that single local environment. So your postdoc who's busily working away on their bioinformatics scripts will be developing those scripts on that local server, installing dependencies and libraries and software as they need it, and creating an environment that is effectively not reproducible anywhere else. So you can document what you do in a bioinformatics analysis when you publish a paper, but the chances of somebody else being able to reconvolute that same computation environment to reproduce what you've done is basically zero, right? So we have a problem, and that problem means that by default, if we develop our software in that way, if we develop our environments in that way, we're creating a situation where bioinformatics is less reproducible than wet lab methods. And that's a problem for me, because I don't like the idea, and I think that we should be doing things better, because you would have thought with all people who are interested in bioinformatics, interested in the technologies that are out there, you'd think that you would try and make use of the available platforms to overcome that problem. And so just as a little illustration of the size of the challenge of how you reconvolute a pipeline, so this is just a little animation from some cholera work that I did at the Sanger. So it's a pretty image with a series of waves of cholera emanating from the Bay of Bengal over the course of about 70 years. Whole process of analysis steps to get to this stage. And the pretty picture looks nice, and a lot of PIs would just see the pretty picture and that's fine. But actually, when you start to think about the number of processes involved and what you need in terms of the compute capacity, you start to realise that this is a bit of a ticking time bomb. So in that sample data set, there's about 320 samples. Each sample, if you uncompress it, is sort of possibly up to 600 to 700. In total, that's around 600 to 700 gigabytes of uncompressed data. And if you think about the pipeline that you go through when you start analysing bacterial samples, chances are your first step will be to do a de novo assembly and a sequence mapping. Okay, so that's 320 jobs sequence assembly, 320 jobs of sequence mapping. And those jobs can be quite memory intensive. So this creates a problem for people who like HPC systems, because HPC systems are designed with certain expectations of what the workloads are going to look like. And the normal expectation is that you'll use between 2 and 4 gigabytes of RAM per core per job. So the sequence assembly there violates that assumption straight away, which means that you're never going to be high on the list of people who the HPC team like. You then take that data and you then analyse it. So you might do some phylogenetics, or you might do some screening for antimicrobial resistance genes. And again, you've got two very different workloads. So the AMR screening might be a whole series of small blasts, so small memory, relatively rapid sort of churn and burn. Whereas the phylogenetics, if you're doing something like RAXML and you've got a diverse data set, you could be using quite a large amount of RAM in a single job, which you can parallelize. And then finally, at the end, if you do something like BEAST, again, there are ways to improve the efficiency of that. There are a set of libraries that are quite useful for that. But again, you've got a set of quite interesting requirements in terms of what you need computationally. And so in an IT sense, when your HPC team design your local cluster, they're looking for use cases to help inform how they design that. And as biologists, when we buy a server, we don't really think about that. But those first processes are what we call high throughput computing. So in that case, you've got lots of jobs running simultaneously, and you don't care how quick each job is. You actually care how quickly you can get all of them done, all the jobs done. The BLAST job is what you call classical high throughput computing. So that's lots and lots of small jobs that all run nicely in serial or can be run in an embarrassingly parallel way. Phylogenetics, large things like XML, they're what you would term maybe more of a classical HPC problem. The code is written in something like C, it's reasonably rapid, and there are lots of ways you can actually speed that up, and that runs quite nicely on an HPC system. And there's something like Beast is possibly HPC, and I say that because if I say that a program that runs in Java is HPC to someone who does HPC, they tend to get quite irate about that. So it's possibly HPC. There are libraries there to improve the efficiency of that, but it's not really there. And so what you've got there is you've got a set of workloads that cut across a wide variety of infrastructures that you would need if you wanted to support all of these. And that's pretty difficult to do. And each one of these analyses requires a whole host of scripts and a whole host of sets of software packages, which mean that if you wanted to reproduce that work from the information that's in the paper, it's going to be extremely difficult to do so, just because of the complexity of that pipeline, those steps required to generate that single pretty picture at the end. And you can go up to the other end of the scale. So this is Nick doing an assembly on one of our large memory machines, and so it's just the output of top. And you can see at the top it's using 1.8 terabytes of RAM. This is the large metagenomics assembly. And again, this is rather than having sort of a whole slew of sort of 4 gigabyte RAM jobs, at the other end of the scale in metagenomics, we can be requiring extremely large amounts of RAM to do what we want to do. So the problem that we have is that as microbiologists, we require a wide variety of systems with quite differing requirements in terms of the workloads we want to put through. Doesn't normally fit with our local systems. Because of the software that we use, it's also hard to reproduce what we do across different systems as well. So, basic premise behind Climb. Wouldn't it be great if there was a single system that microbiologists could log onto, which would support a wide range of different workloads and would allow you to share data and to share the software approaches that you've got? That's a pretty simple idea. It's a pretty simple observation. Data is more easily shared, especially when there's lots of it, if you're in a single environment and everyone has access to it. And software is also more easily shared on a common platform. And a common platform can also can bring all that together, and it can do so easily. So again, thinking back to my Sanger days, Sanger has a well-designed system. They understand their workloads very well. They do lots of benchmarking, lots of testing, and they've designed a system that fits the profile of what their users want to do extremely well. And because it's a single system across the institute, it provides a single environment for sharing data and for sharing software. Okay, so across the institute, there's opportunities for collaboration and for people to use things that everyone else is doing, and that works brilliantly. And I think that environment doesn't get the respect it deserves, because I think that having that fundamental there actually empowers a lot of what people like the Sanger do. But the problem is, for most of us in universities, the chances are you're not going to have a critical mass of microbiologists, of people requiring that sort of a system in a university for the university to want to spend money on building that system. So the problem is, most of us, I guess, we're in departments where there might be a small number of microbiologists locally, and we don't have the critical mass or the power within the university to get them to build a sort of system like that locally. And so what we thought, and I guess the foundation of the project, was that we could, if we were to join across universities to create a consortium between multiple universities with multiple microbiology groups, we could create a platform that would be free for anyone to access around the UK. And that this would then get around the problem of not having enough people locally, and would actually create opportunities for collaboration and data sharing that go between institutes, which would be quite nice. So you're going to be using a thing called OpenStack today, you won't necessarily know it because that runs underneath. And how you achieve that is through virtualization. So virtualization is basically a way of running multiple distinct servers on the same physical hardware. Okay, so you have the server. If you could take everything in an operating system and containerize that in a large single file, and then you could have a piece of software which could boot that as if it were a machine on its own. That's the idea behind virtualization. So virtualization is a mainstay of enterprise computing. So companies use this all the time. And they use it because what they recognized was that you could have a number of large servers which you could share up in small slices, and you could then much more efficiently manage your server estate through having a smaller number of larger servers which could run a whole series of processes at the same time. It increases reliability. There's a whole slew of reasons why virtualization is widely used in business. And actually it fits our needs very well too. Because in a virtualized world, you can containerize an operating system. So when I'm installing software onto a server, and I'm installing a whole host of different dependencies, and I get my software working, rather than requiring someone else to reconstitute that whole development environment, if I could just share the whole machine, and somebody else at a single click could spin that machine up and access it, they can literally pick up where I finished off. Okay, so virtualization is a technology that allows that to happen, and it makes it possible. And so that actually potentially answers a lot of the questions around bioinformatics in terms of reproducibility, because if you share a virtual machine with all the software pre-installed that you've been doing your analysis on, and I guess the ideal world is you're analysing a set of data, and at the point that you come to publish, you take a snapshot of that machine and a snapshot of that data and share it, then at that point someone can literally reproduce exactly what you've done to generate the results in your paper, which is quite an interesting idea. So, ideal world, user wants a server, user could be from anywhere, and you want a system which will spin up a VM, find an available server, slot that onto the server, and use up that chunk of the server just for that single user, use as a single point of access. Then what you can have is other users then come along and want to do the same thing, and they are each also given a piece of the server to use. So they have this silo of their own, which is guaranteed resource for them, and is separated from everyone else, but is done in such a way that it can then be shared subsequently. I think that's quite an interesting idea. Not the only one. That concept is central to systems like AWS, Amazon Web Services. It also underpins a huge number of services that we use every day, and that includes things like Google, Netflix, Dropbox. These services are all underpinned by this same concept of virtualization. So a large infrastructure of servers using virtualization to define the size of the server that's appropriate for the job that you want to use it for. And so it means that you can leverage, use a business term, commodity servers that are quite cheap to buy and cheap to run, and you can use them in all sorts of ways. And then if you're a company, it means you can sell off access to small slices of those servers and make lots of money. So Climb was the same sort of concept, to provide a service for the UK microbiology community where you can get access to a reasonably large amount of compute on a shared infrastructure, and just without the profit, I think. People do say sometimes, why don't we use a commercial? Why not just use a commercial cloud? So informatics workloads can have requirements that require lots of RAM and a good number of CPUs with a lot of storage. And the problem is that the users that commercial cloud providers are targeting really aren't the sort of users that bioinformaticians are. So what that means is that the price for us is higher because we're not the users they want on their system. And there are also some pretty big limitations in terms of how big you can go with VMs on things like Amazon. So I think the biggest Amazon VM is about 250 gigs of RAM. So if you wanted a really large VM beyond that, then you're going to run into all sorts of issues in terms of being able to do that within their system simply because it's not provided for. The costs of storage are also high. So 1 T of storage on Amazon, on S3 on Amazon Standard Storage System, costs about $30 a month. And our current costs on Climb work out to be about £3 a month per terabyte. So there's a big difference in cost as well. So if we wanted to do the same thing, if we wanted to take Climb, the system that you're going to be using, and offer that just out via Amazon tokens, you get a lot less for your money than actually we've got by doing it ourselves. It's entirely possible that in the future, things like Amazon, things like Google will actually be of better value than doing it locally. And at that point, you still have the benefit if we're building up a repertoire of images and libraries of data, we can still run that on something like Amazon. But we built that resource ahead of that time, which is there's also some benefit there. So some of you may have been at Built-in Bioinformatics, which was the first time that I talked about what we were doing with the system. And we had this set of aims, which we mostly met. So to create a public-private cloud for use by UK academics, so public in the sense that if you're not part of Cardiff University, Warwick University, Birmingham University or Swansea University, you can get access to the system. But if you're not a microbiologist, then you can't, so it's private to an extent. We wanted to have a set of standardized pipelines, standardized images on there, which would already do a lot of what most microbiologists would want to do. And we wanted to have this big storage repository underneath. And so we had this sort of expected specifications. So I guess the key ones being that we wanted to be able to support at least 1,000 virtual machines simultaneously. We wanted about 4 petabytes of object storage, and we wanted some local storage as well. And so in terms of where we are now, the system as it stands grants access to PIs from any organisation where you have a .ac.uk, .nhs.uk or .gov.uk email address. And then those PIs can choose to bring in people, collaborators from outside to have access to the system. We already have a set of standardised cloud images. You're going to be playing with at least one of them this afternoon. And we have this storage repository now up and running. We're working with people like the EBI to provide access into other databases like the European Nucleotide Archive. And we have the option now also of supporting orphan tools and data sets. So we're going to introduce you today to the first set of VMs on the system. Hopefully you've all got, or you will all have by the end of this session, accounts on Climb. You'll have VMs running and you'll be able to use the system. It's been a lot of work, but it's hopefully going to be worth it. So in terms of the system itself, we have 4 sites connected over Janet. We have over 1,000 virtual machines simultaneously. We don't do a thing called oversubscription, which means that when you spin up a virtual machine with 8 virtual CPU cores, that's what you're getting. So what you see is what you get. Some providers, for example, I don't know if Amazon do, but some cloud providers will actually do oversubscription where they'll say that they'll give you 8 virtual CPU cores, but actually it's a contention ratio of say 1 to 16, so you're getting a lot less. We managed to procure 7 petabytes or so in total of object storage, and so when you replicate that, the total amount usable comes down, but we've got a reasonably decent amount of that. We also have 400 to 500 T of local high-performance storage, so we managed to get beyond both of those. You're going to be introduced to Brin, which is our single system for logging on, and we're also working on getting between-site data replication fully up and running. And we've also designed the system to hopefully enable other universities to be added. So we have 4 sites. We're running this system called OpenStack, which you can read about or ask about today. Procured the hardware doing a 2-stage process. Our compute for OpenStack is provided by OCF. They were the integrators, and the hardware is IBM Lenovo. And Dell and Red Hat provided our object storage. That 7 petabytes of object storage is a thing called Ceph running on top of Dell hardware. And then we have networks all provided by Brocade. And we also now have a fairly clear reference architecture, which means that we can share that with other universities, and if people want to try and do the same thing or want to plug into Climb, they can do so. In terms of the actual what the 3.6 million of funding bought, we got 8 router firewalls, so 2 per site. Each one can do about 80 gigabit of routing in theory. We have 12 OpenStack controller nodes, 3 per site. We have 84 64-core 512 gigabyte RAM compute nodes, 12 192-core 3 terabyte RAM large memory nodes. We have about 2 petabytes raw of GPFS, and that's locally distributed between the sites. We have 4 controllers per site. and that's for providing some sort of local scratch storage. And we also have about 7 petabytes of Ceph, and that's 108, I think, 64T nodes running on the system. In terms of performance, it's generally pretty good. So we've benchmarked it against Microsoft Azure and Google Cloud, and so what you see is the Microsoft Azure A8. In the middle we have Raven, which is the Cardiff University HPC system. A score of 1 is identical to Raven. A score of greater than 1 is faster than Raven. A score of less than 1 is slower than Raven. Raven's about 2 years old now. What we found was that the climb, so CFCL XL Ubuntu is a standard Ubuntu image running on Climb, doing a benchmark again across a series of bioinformatics workloads. And what you see is that basically we're better than a top-of-the-line Microsoft compute instance, which is incredibly expensive in comparison to what we're able to offer. Also on the far end, and just to make a point, you'll see the benchmarking performance of an Office desktop, which was the fastest of the lot. And so that's just the point. So that's an Office desktop with about 16 gig of RAM, a solid-state hard disk, and a 4-core Xeon processor. And the simple fact of the matter is that there's a lot of workloads that you can do quite happily in bioinformatics, which you shouldn't necessarily be doing on a big, expensive resource. And so there is a certain point to thinking about what you're doing, because actually you'll get much better performance running things locally in that sort of situation compared to running it on a cloud. But in terms of our performance, we're pretty happy with where that is, and we think it's actually quite good value for money. So what it means for you, so ideally we don't really provide small machines. The small machines we provide are 4-core and 16 gig of RAM, and that's about the minimum. And they're really for maybe more for training than doing serious work. So our personal servers start with 4 CPU and about 15 to 16 gig of RAM. Our group servers are maybe more where people would be looking at using these, I guess, in anger, and they're 8 CPU cores and 60 gig of RAM. We have other flavor sizes available on request. So if you have a specific requirement for maybe slightly more CPU or slightly more RAM, it's worth talking to us, because we can possibly do something. It means in terms of researchers, it means you get free and immediate access to dedicated hardware to do your data analysis. And so, sort of in comparison, in terms of the value of that freeness, a personal workstation with a spec that's similar to our server retails for about £1,000, and a standard server with about 8 cores and 64 gig of RAM, that can be maybe 3K or so. So that's the sort of value that you're getting from this system that's giving you free access to that kind of hardware on demand. So what we do is we register each PI, and each PI is given what's called a tenant, and I'll mention a bit about tenants when we get on to talking about OpenStack. But what that is, it's a project with a fixed quota. So that means each PI's group that's been registered has a quota with a default allocation. So 640 gigabytes of RAM, up to 10 instances, 20 volumes, a total disk of 10 terabytes, and a maximum of 128 cores. So that's a reasonably substantial amount of compute that's available. Now, it's up to you to manage how you're going to use that, and there is a distinct possibility that we're going to run out of space at some point. There is a proviso that if you spin up VMs and you leave them sitting there doing nothing, we're going to know about it because we can monitor the system, and we will probably suspend those so that other people can get access to it. So just something to bear in mind. But in theory, we have the hardware and we have the quotas actually for people to be able to do quite a lot of work with what we've got. In terms of system access, so we had about 50 beta testers over the last year who've all been using the system, trying it out, and we've had a lot of feedback from that. And from that, we've developed a couple of service offerings. So we have 2 methods for access. So Brin, which you've all been introduced to, is our system for logging in and registering yourself on the system. And you can launch virtual machines through Brin, and you can control them through Brin. And that's our standard mode of access for pretty much, I guess, 95% of people are going to want to go that way. We also have, as part of the OpenStack system, a dashboard for controlling other aspects of virtual machines, for spinning up new virtual machines, for controlling volume creation and doing things in a much more precise way. You probably don't want to play around with that, because there's a chance, if you do and you don't know what you're doing, you could destroy your data or your virtual machines that have been launched from Brin. But if you want to generate new virtual machines, or you want to generate much bigger virtual machines using your quota, you can actually access that as well. So we have two options for access. All users on the system are part of a project, and that project is a group, so it links to a PI's group. Project owners are principal investigators, and it means that a PI has to register first, and then the PI has the ability to send out invites to members of their group to get access to that project. Now that might seem a pain. I mean, the registration process is actually pretty short, but the problem that we've got running a service is there needs to be clear lines of responsibility. We're giving you the opportunity to spin up a virtual machine, and that virtual machine could be used to do anything, right? And obviously there are lots of things you aren't allowed to do. Some of them are legally not allowed to do. Others are based upon the usage regulations for the universities. So that goes without saying that you shouldn't be hosting pornography on there. You shouldn't be hosting illegal images. You shouldn't be. You shouldn't be hacking other sites. You shouldn't be Bitcoin mining. But it's much easier for us to manage that and to manage postdocs and PhD students through the system if they're accountable to their PI who runs the project. So that's why. That's one of the major reasons for doing that. The nice thing is as well, so this system allows us to bring in overseas collaborators where they're doing work that's relevant to UK-funded research, and by monitoring registrations by PI, it actually gives us a nice mechanism to track the impact and track how the system's being used. So in terms of system status, you're mostly going to be using Warwick today. So obviously we have 4 sites, and so the VMs that have been spun up on Warwick, which is the default region at the moment. Birmingham's online. It's working and it's freely available. You can spin up VMs there if you want. It will run out of IP addresses at some point if lots of people do that. The reason you might not want to run things on Birmingham is there was a bit of an incident in the data centre a week or 2 ago, and there's going to be some remedial work that has to be done this weekend, so everything's getting switched off. So that's why you might not want to use Birmingham. Cardiff's online. It's available. We haven't fully stress tested that yet. We've been stress testing Birmingham and Warwick over the last sort of month or so, and Cardiff's fully working, but it's not been stress tested yet, so you might get some slight instability if you use Cardiff. And the last thing that we're waiting for at the moment is to do the integration of the Swansea system with Brin, and that will be happening hopefully over the next few months. There are other parts of the project as well. So we have a forum. So if you go to your Brin login page, there's a community tab. You can click the community link, and there's a forum where we encourage you to post and participate. We have a Twitter account that you've probably come across, so please engage with the Twitter account. And if you have particular papers that you've published where you've used Climb, tweet the Climb account, because we'd like to know. For those of you who are on the Google group is now dead, and the communication for the project will be going through the Twitter account and through the forum. Setting up the system has been sort of a 2-year endeavour in terms of buying the system and getting everything in place. Our next part of the project isn't just to run a service. We're going to be offering out training events, and we're going to be looking to work with people who are currently delivering training events to really maximise the impact and the reach of the project. So look out for those in future as well. And if you've got an interesting idea for training that you're going to do, please talk to us, because it might be that we can give you free resource on Climb to actually run that event. So today we're introducing the system. We want you to use the system. We've put some tools and some software on there already. But the thing I want to emphasise is, so the grant looks pretty big in terms of the amount of money that was with it, but the actual grant doesn't have much money in terms of software development for developing pipelines and for putting software onto the system. So we're sort of relying on people in the community to contribute in terms of putting their pipelines, putting their workflows or their virtual machines onto Climb so that other people can make use of it. So we want your support there. We also want your support in terms of RC UK and getting funding. So, I mean, if you find that Climb's really useful, thinking about ways that we can get more capacity in terms of developing our the tools and resources that are available on Climb is something that's going to be really important for the longevity of the project. And so that's something we're happy to talk about with people as well. Might be a few teething problems today. This is the first time we'd have had so many users using the system simultaneously. So when things break, we've got Radoslav, we've got Marius, and we've got Matt, who will be circulating to help with serious problems, and then the rest of us will hopefully be around to help you with questions around the tutorials and things. In the next couple of months, there will be a Climb paper coming out. I think it's going to be put up on a preprint site in the next week or so, hopefully, and we're going to be sending that out to a journal to review ASAP. When you use Climb and use it to analyse your data, please reference our paper and please acknowledge us, because that's pretty important in terms of realising getting an idea of what the impact is for this system. We think the system's going to be really useful for a lot of people, and that requires then, if people in the community find it useful, to acknowledge that, so that then we can go back to people like the MRC and say, this is the benefit of having the system, please carry on supporting it.